नंबर कौन सा आरएनए? एमआरएनए? नहीं नहीं, एमआरएनए बहुत कम होता है सेल्स में। एमएससी स्टूडेंट्स हो, राइट? आरएनए पढ़ाया गया है क्लास में? कौन सा? बाय नंबर, टीआरएनए। ओके ओके, नाउ डीएनए से आरएनए बन रहा है। सो फ्रॉम ह्यूमन जीनोम, व्हाट इज द परसेंटेज ऑफ जीनोम दैट इज गिविंग राइज टू आरएनए? 2%? इतना कम नहीं हो सकता, नहीं। 10%, वो भी बहुत कम है। इवोल्यूशन कैन नॉट बी दैट स्टूपिड, राइट? जस्ट 10% बनाने के लिए बाकी 90% जंक डीएनए डालेगा इवोल्यूशन। हाँ, कोडिंग पार्ट, बट मैं पूछ रहा हूँ ट्रांसक्रिप्शन। कोडिंग, जो कोडिंग नॉन कोडिंग है, दैट इज फॉर आरएनए। आई एम टॉकिंग अबाउट इन टर्म्स ऑफ योर होल जीनोम। 75%, actually more than 75%, that is transcribed. Okay, so this is your DNA, right? How big is the genome? 6.6. okay, हाँ, close. okay, वो diploid का. okay, अब हाँ, actually it is 3.2 billion. so ये कितना लंबा होगा? 3.2 बिलियन, 2 मीटर। हाँ, ये सबको याद है। हाँ, ओके। तो अगर एक इंडिविजुअल से निकालेंगे डीएनए, कितना बनेगा? कितना लंबा होगा? 2 मीटर एक सेल से, फ्रॉम वन इंडिविजुअल। बोलो, गलत होगा तो मैं मार्क नहीं काटने वाला हूँ, ठीक है? ओके, एंड यू नो हाउ लेंदी दैट इज? ओके, so that big DNA you have, and 75% of it is getting transcribed. So you have huge amount of RNA, right? So DNA ट्रांसक्रिप्शन टू आरएनए, ये आई थिंक बहुत अच्छे से कवर हुआ है, ठीक है? और ये जो आरएनए बनता है, कितने टाइप के आरएनए हैं उसको बिना देख के बोलो। एसआईआरएनए? नहीं 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 नहीं, एसआईआरएनए नहीं है। एसएचआरएनए सेल्स में नहीं होता है, एसआईआरएनए भी सेल्स में नहीं होता है। Those are exogenous RNA. We put them into the cell. miRNA, then siRNA नहीं, siRNA is exogenous. We make it outside and put it. और snRNA, long non-coding RNA, और SNO RNA, right? SNO RNA. So the RNA actually discovery started from 1950s, okay? And with time, people started discovering rRNA, mRNA, then link RNA. GIST, H19 are the long non-coding RNAs. Then slowly you have lot more RNA, and then when the RNA sequencing, next generation sequencing came, people found tons of different kind of RNA, like mostly non-coding RNAs, because they don't code for protein. So उसके बारे में ज़्यादा रिसर्च हुआ नहीं था. So now in last 20 years, 25 years, people have started looking at all kind of RNA. They are not coding for protein, but they are important for the cell. Okay, cell physiology or disease. Okay, so if you look at it, so your 76% human genome is transcribed, giving rise to RNA. From that 100% RNA, 98% is actually non-coding, 2% is coding. Okay, and then the non-coding part, if you divide them into two halves, like long non-coding RNA and short non-coding RNA. Long non-coding RNA are basically RNA, those are more than 200 bases. Then short are less than 200 bases. So in the long non-coding RNA part, you have many RNAs like intergenic, antisense, sense, intronic RNA. In the small one, microRNA, pvRNA, small nuclear RNA, snRNA. So those are the RNA. And if you look at how much they are present in the cell, like I already mentioned, by mass, rRNA is the most. Okay. And then tRNA, then mRNA, then other non-coding RNA, link RNA. So microRNA is very negligible. So it is like 0.00 some percent by mass. Okay. And if you run the RNA on a gel, which RNA you will see? rRNA. Yeah, of course, that is the most abundant one, correct? So you will see mostly rRNA and then little bit of tRNA. As you can see here, 28S, 18S. This is tRNA. MicroRNA you will not see actually, but that comes around that position. 5S rRNA, tRNA. You will see those on the gel. You will never see mRNA. Like many times in interview, people ask you what you see on the gel if you run. Most of the time people say mRNA we can see on the gel. You will never see mRNA on the gel. Okay, even if it is like 10% of the total RNA, you will not see that. Okay, why? Bolo.
Few number of mRNA present in there.
Not few. By mass it is 10% around, right? But still you don't see them because the size is not same. For 28S, 18S, the size is same. So everything comes at one place, you can see that. But if something is like 100 bases, 200, 2 kb, 5 kb, so they are all over the gel. You will never see them. Okay. And by number, you see how much tRNA you have. It's way more than your rRNA. Okay. And rRNA is the second most abundant one, then followed by other RNAs. Okay, 7SL is a link RNA, snRNA you have little bit, then SNO RNA, mRNA population by number is very little usually, but by mass it is more because the size is bigger. For rRNA it is the same. By number they are less than tRNA, but by size they are much bigger than tRNA. What is the size of tRNA? rRNA? नहीं पता. Okay. So the 28S है, that is around 4.7 kb. Okay. 18S is I think 1900 bases, and tRNA is 85, right? 85 bases. Okay. So, and when we talk about function, the mRNA is the most functional thing we always consider because that codes for the protein, right? rRNA and tRNA, these are integral part of the protein translation machinery. Without them, protein translation cannot happen. Then snRNA, SNO RNA, these are involved in the splicing. Okay, so without them, your mRNA will not be formed. You will all be having the pre-mRNA, so no splicing is happening. And then the regulatory RNAs, microRNA, link RNA, and circular RNAs. These are regulatory RNAs. Anyone know circular RNAs? No? कोई नहीं? लूप होता है. Okay, we'll see that. Okay, so link RNA. How many of you know link RNA? Functions of link RNA? Long non-coding RNA? नहीं पता. Okay, so these are RNA without any protein coding ability. So अगर कोई RNA है वो प्रोटीन कोड नहीं कर रहा है तो क्या करेगा वो? सेल में बन गया, सेल में है. ओके, इट विल डू समथिंग, राइट? क्या करेगा? बाइंड विथ प्रोटीन और डीएनए, और किसके साथ बाइंड करेगा? खुद के साथ भी बाइंड करके सेकेंडरी टर्शियरी स्ट्रक्चर बना सकता है, करेक्ट। देन माइक्रो आरएनएस रेगुलेटरी है, उसको भी बाइंड कर सकता है, बिकॉज माइक्रो आरएनए टारगेट एमआरएनए। सो माइक्रो आरएनए को थोड़ी ना पता है कि ये लॉन्ग नॉन कोडिंग आरएनए, सीक्वेंस कॉम्प्लीमेंटरिटी है, दे विल आल्सो टारगेट, राइट। So it can interact with mRNA, regulate translation, it can interact with microRNA and sponsor the microRNA. MicroRNA usually inhibit the translation. So if you have the long non-coding RNA that interact with the microRNA, so microRNA activity is inhibited. So it will no longer inhibit the mRNA translation, okay. And then it can bind to the DNA, okay, and then regulate the transcription. It also activate transcription or inhibit depending on the interacting partners, the cofactors it is interacting with, right. And this is microRNA, okay. So anyone knows what microRNA does? Translation inhibition, right? हाँ या ना? नहीं, हाँ, mRNA degradation और okay. Mostly microRNAs in animals, they inhibit translation, not degrade, okay. In some cases they degrade if the mRNA is perfectly complementary to the microRNA, and that act as siRNA. But in plants, mostly microRNA degrade the mRNA, okay. But in animals, remember, it is mostly inhibition of translation, not degradation. In some cases it will degrade, but most cases it will inhibit the translation. And how it does is, either it will when mRNA translation happens, the cap and the poly A tail, they interact with each other, they make a loop, okay. So the microRNA binding inhibits that loop formation, so that the efficiency goes down. Or sometimes the microRNA binding inhibit the movement of the ribosome, slow down the process of translation. And then the last one is like degrade. If the mRNA is degraded, so you will have no protein, okay. And then the other one is the circular RNAs. So nobody knows about this, I think. हाँ या ना? No, okay. So this is the RNA actually my lab works on. It's a new kind of RNA without any 5 prime or 3 prime ends. So as the name says, it is a circle. So they come from any pre-mRNA. When the splicing is happening, so when your exon 1, 2, 3, 4, they join in a linear fashion, you get the mature mRNA, right? If the suppose exon 2, end of the exon 2 joins the beginning of exon 2, or end of exon 3 joins the beginning of exon 2 or 3, then it will be a head to tail ligation. So that makes the exons as a circle, okay. So in that case you will never have the 5 prime or 3 prime ends, and because of that they become very stable because they cannot be degraded by the exonuclease. So in the cell, if the circRNAs are made, they will stay for longer time. The half-life is much more than the linear mRNAs or long non-coding RNAs. Okay. Yeah. What is the reason? Reason I don't know, but they are there for the regulation. So they don't, most of them don't code for the protein, but when they make a circle, they're very stable. So they can act as a sponge for RNA binding protein. They can sponge the microRNA. So because of their stability, they can stay longer, and then they can regulate. If they bind to something, that will remain for a longer time. So for regulatory purpose they are made. The number is huge, but all of them may not be actually functional. Okay, but some of them are very abundant, and in humans we have like more than 1,000,000 circular RNA discovered from RNA sequencing in last 12-13 years. But a few hundred have been characterized, not a lot of them, and some we are characterizing in different systems in my lab. We will see that. So as I said, they act as microRNA spawns. They bind to the microRNA, they bind to the RNA binding proteins. Either they regulate some cellular processes by binding to some splicing factor translation regulators, and some circRNA have been reported to regulate transcription by binding to the transcription factor and snRNA, and recently they have been categorized, some of them as coding circular RNA, so they can translate into protein. As I said, the mRNA prefers to translate in a loop manner when the cap and poly they are joined. So now you have RNA that is a circle itself. So if the ribosome is moving, it will be easier for the ribosome to move around. But the start is always difficult because you don't have cap. Cap is always required for efficient translation. But there are elements like methylation, or there are IREs, internal ribosome entry sites. Those facilitate the ribosome sitting on the RNA and then scan for the starting. So once you have the ORF starting, it will be moving round and round. So it will have more effective translation for a longer time because it is very stable. So now people are trying to use this for therapeutics, for the gene therapy, because it is a stable molecule. Okay, so let's go back to the RNA modification. Now we know different kind of RNA molecules and their function, right? So how the modification regulate their activity, right? So in DNA, what were the modifications you learned in last 5-6? Methylation, धीरे 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 मुझे सुनाई नहीं दे रहा है. हाँ, मिथाइलेशन, देन एसिटाइलेशन और phosphorylation. Chromatin remodelers are proteins. So that is not DNA modification, but that is protein modification that regulate transcription. Okay. Any other modification? Ubiquitin. That is also protein modification. Or for DNA there are not many modifications actually. जो भी फिर प्रोटीन रिमॉडलर प्रोटीन पे जो ज़्यादा मॉडिफिकेशन होता है, DNA is not that much modified, but when it comes to RNA they are heavily modified. Okay, because DNA, if they are modified and your transcription goes wrong, it will be very disastrous for the cell. So you cannot like modify the DNA with anything. Okay, so evolution didn't prefer that, but for RNA you have hundreds of modifications. Okay, because of their diverse function, like they can change the stability, they can change the structure of the RNA, they can regulate the translation of the RNA, okay, and the interaction with other molecules. So that's why RNA modification is like you have hundreds of them. Okay. So 170 modifications. This is like a old data, 170. It is actually more than now close to 300 modifications have been reported. Okay, and this is very critical for the function of the RNA. Any of the RNA modification you guys know? Splicing. Splicing is basically the splicing of the introns out of the mature RNA. So that is not modification, but modification plays a role in splicing. Okay. Poly A tailing. Okay, so that is not actually modification. It is what you call that? It's synthesis actually. It's not modification. So when we say modification means the nucleotides are modified. Okay. In poly A tailing, you are adding poly A tail. Okay. So that is co-transcriptional. Just a cap hai, that is a modification because that is not a regular nucleotide, right? What is the cap structure? हाँ जीटीपी, बट क्या बोलते हैं उसको? 7 methyl. हाँ, so that is a modification. So that is not a regular G, right? And it is joined 5 prime to 3 prime या उल्टा है? उल्टा होता है, right? Sorry. हाँ, तो 5 prime cap. So that is one modification. Then anything else? Methylation है ना, right? So methylation is one of the most abundant modification on any of the RNA. Okay, whether it is mRNA or non-coding RNA, any of the RNA. And the first, here, this is a database, MOD omics. Okay, so modification omics. So that lists around like over 300 RNA modifications. So this is a database, and the first nucleotide modification was discovered in 1957. And which one was that? Pseudoiridine. Okay, it is not methylation. Okay, pseudoiridine, but that doesn't happen in all cells, all kind of RNA. But M6A methylation happens in all kind of RNA, and that is the most abundant one. Okay. Because this modification changes the stability of the RNA. If some modification can stabilize the RNA, they are a potential candidate for being used in the RNA therapeutics. Okay. So the major types are M6A, so N6 methyl adenosine, that is the most abundant one, then pseudoiridine, then your M5C and A2I. So all of this, if you look at it, they either alter the structure or the stability of RNA, or they regulate the translation of the mRNA, in which they are like modifying the mRNA. Okay. You all don't need to write everything. Okay. It will be on YouTube, right? So you can always see the slides. Okay. And how does this happen? M6A, I said like it is the most abundant modification, right? So which enzyme makes DNA methylated? Methyl transferase. कौन सा नंबर? DNA के लिए? हाँ, मुझे भी नहीं पता. हाँ, 1. Okay. Something else? I don't know. Okay. And for RNA, metal 3, 14, 16, WTAP, and there are a few more in the other slides we'll see. And if something is modified, someone need to recognize them, right? So these are the readers. So the topic name is RNA modification and reader. So if the RNA is modified, someone need to read them. Okay, read them means bind to that modification. So where is that? Yeah, this one, YTHDF. So that is a reader. There are many more in the next one. We'll see, next slide we'll see. And the erasers, if something is modified, so something can reverse that back, okay, because every time you don't need that modification. So depending on the cellular physiology requirement, cell modifies and sometimes they reverse it back. So those are the erasers, so they will remove the modification. For M6A it is FTO and ALKBH5. So these are the two major players who can remove the modifications. Okay. And for this pseudouridine, so this was the first one that was discovered for the modification, and this is very important for rRNA and tRNA. Okay, many of the nucleotides are modified with pseudouridine, and these are the modification, and with the modification the nucleotide position changes, and the what you call that, like the base pairing, when the nucleotide moves, the base pairing also changes. So because of that they regulate the structure and the stability of the RNA. And the major RNA types like tRNA and your link RNAs and rRNA and mRNA, which RNA you think is the most modified one? rRNA. Kyun? Need to be more stable. Okay. Because of the abundance. tRNA. तो चार ही ऑप्शन है, तीन ही है बेसिकली, राइट? तो कौन सा होगा? tRNA. हाँ, actually it is tRNA. It's highly modified. Okay. And then rRNA, mRNA also, but compared to tRNA, there are very less modifications. But tRNA is heavily modified. Okay. And these are the major modifications on rRNA. Okay, 91 pseudouridine, 105 2-oxymethyl, and 10 methylated bases. Okay. Yeah, these are some other modifications on rRNA. Okay. So then when we go to tRNA, 15 to 25% of the nucleotide are modified. Okay. So you can imagine how heavily some RNA can be modified, and any modification on tRNA can affect the translation, because any modification we are talking about, they have effect on the structure. First thing is the structure of the RNA, and then the tRNA is important for what? Amino acid synthesis, because of its binding to anticodon. Okay, so if you imagine there is a modification on the anticodon, तो क्या होगा? It will not bind, or it may bind better, it may be recognized better, right? So both things are possible. So, and the other loops in the tRNA, they are also heavily modified. So they are also important for the amino acid joining or the structure to recognize the ribosome, right? So any modification on tRNA can affect its ability to bind to the anticodon, first thing, then the structure, which is important for binding to the ribosome, and then the amino acid joining to the tRNA. So all these processes are very much essential for any cell. So any modification is very crucial, like it can enhance, it can also like inhibit the translation. So now you see the modification on tRNA. So you see like half of them are the modifiers, and these are the enzymes they do those modifications. So it is one of the heavily modified RNA in the cell. And then comes to the mRNA. Okay, it is not that heavily modified as you see for the tRNA or rRNA, but it is modified. Okay, and the first major modification is the cap, 5 prime cap, because that happens as soon as the RNA is synthesized. Then the other modifications, 2-oxymethyl, then this M1A, M6A, pseudoiridine, and M5C. So all these things are added. So you know 5 prime cap, 5 prime UTR, coding region, 3 prime UTR, poly-A tail, all know? Okay, what is the difference between coding region and exon? Coding region is the compilation of exons. Okay, and exons? What is the full form of UTR? Untranslated region. So CDS is the coding part, translated region, and the start and the end are the untranslated region. And exons? So इसमें exon कहाँ है? Coding region. Okay. Anyone else? नहीं. एक्सॉन और इंट्रॉन के बीच में डिफरेंस क्या है? कोडिंग नॉन कोडिंग नहीं. हाँ, not expressed means spliced out, right? So exon, intron, both are part of the pre-mRNA. Okay, when the gene is transcribed, they are transcribed into a whole transcript. Cell doesn't say this is intron or exon. When the splicing machinery comes in, they splice out some portions out of the RNA, and others are joined together to form a mature mRNA. So whatever RNA is left after splicing, that is your mature mRNA, and whatever portion of the pre-mRNA it contains, those are different exons like 1 to 10, and उसके बीच में जितना unwanted sequence था वो निकल गया, but that has nothing to do with your coding or translated or non-translated region. So everything you see from cap to poly-A tail are exons. Okay, so then when the exons are joined, you have a full length RNA. Poly-A tail is also added, so that is called mRNA. When the mRNA comes out of the nucleus, then this business comes, whether something is UTR or coding region. Before that, it is all exon. From start to end, it is exon, and poly-A tail is added, cap is added. Then the translation machinery decides from where to where it will translate. Okay, so that portion is only your coding region, basically like the you call the CDS coding sequence. बाकी जो बच गया वो है untranslated regions. Okay, so most of the time people say exons are the coding regions, right? No, everything is exon only. Then coding and non-coding with respect to the translation ability of the RNA. Okay, so how big is mRNA? Anyone knows? Okay, suppose you have a mRNA which is 900 bases long. Okay, so how big the protein will be? 300 amino acids. Okay, very good. So this kind of things comes in the interview, like, and I didn't tell you where the coding region starts. I only said 900 nucleotide. I never said the ORF starts from 1, right? You should ask where is the start codon. Without that, how do you know you will have 300 amino acids? And where is the end? Okay. And suppose you put that in a circle, okay, then what will happen? If you don't have any stop codon, it will start and then go round and round, right? Okay, so mRNA modifications, you see the writers here, mostly I said metal 14, metal 3, and here you see CMTR1, PCIF1. There are other enzymes, which this is for the 2-oxymethylation, then metal 3, metal 14 is for M6A, and FTO and this LKBH5. These are the erasers. They can reverse the M6A modification, and then decapping enzyme can remove the cap. So when the mRNA need to go for degradation, if the cap is there, it will not allow the exonuclease to degrade. So one function of the cap modification is to protect the mRNA from degradation. Most of the time when the degradation happens in the cell, it is mostly exonuclease activity from either 5' to 3' or 3' to 5'. So 3' doesn't have cap, so it can easily degrade. But if enzyme wants to degrade from 5' to 3', then the cap will block it, right? So the cap need to be removed. Okay, so that removal happens by the decapping enzyme. So once the decapping enzyme removes the cap, the RNA now is susceptible for the exonuclease activity, and then it can be degraded. And the poly-A tail also act as a, it is not a modification, but it is kind of protect the RNA